Genetics and inheritance. We're going to begin by looking at what genetics is. By definition, genetics is the study of genes and how they are transferred from one generation to the next. Your inheritance, then, is the DNA that you've received from an ancestor. Genes are made up of structures called alleles. Alleles are different structural and functional variations of homologous genes. If something is considered to be homozygous, that means that both alleles of a gene are identical. Homo in this case refers to same, therefore both of the alleles of a gene are going to be the same. Heterozygous, on the other hand, hetero refers to different, therefore the alleles of the genes, if they are heterozygous, are going to be different. And this is, of course, the result of years of mutations. A genome, then, is a complete set of genes. And if we speak about the gene pool, that would be all genes and the alleles that make up those genes. So the genomes is going to more so be an individual's set of genes, whereas the gene pool would be the population's genes. A genotype, then, is that complete set of genes or the alleles that we're going to have for each individual. And we'll talk a little bit more about this, but if we're talking about somebody's genotype, we would say that they had a dominant gene or a recessive gene. A phenotype, then, is the observable physical and functional traits. So whether somebody's tall or short, has blonde hair or brown hair, etc. Now, the way that we determine what the probability is that a particular gene or allele will be inherited is something called a Punnett square. So to show you, we're going to start looking at the picture A here. At the top you see that there's a female. On the left-hand side is the male. So since a female has eggs to offer up as her gamete, you can see that at the top we've got one big A and one little a. That means that the female has a 50-50 chance of giving that big A away as the allele or giving the small a away as the allele. So what we do is we separate those into the respective gamete, again in this case female, so it would be in the egg. If we look to the left of the Punnett square, we can see the male gene. The male has a big A and a little a for alleles as well. Therefore, each sperm will either contain a large A or a little a. Now, to calculate the probability, all we do is take our alleles that we have aligned on the top and the side, and we simply move them down into the respective boxes. So here's what I'm going to say would be the easiest thing to do. Looking at the top with the female, take the large A, move it down one. So you can see that first box that's dark purple, it's got a big A in it. Take that same A and put it in the bottom box below it. Now, go back to the little a, bring it down one box, and bring it down a second box. So we simply fill in the boxes with the respective allele. Looking at the male now, we do the same thing, only we bring them across the Punnett square. So if the large A in the sperm gets moved over into the square that's dark purple, we've got a large A given from the man, and then again moving that over into the box to the right of it, we've got a large A as well. Then we would do the same thing for the sperm that has the small a in it. We would simply bring it over to the right into each of the respective two squares. That's going to tell us now when male and female join together what the chances that an individual would have those particular alleles. If you look down to B, C, and D in the picture, they've done the same thing, just with different genes. So instead of the genotype being big A, little a, as in the example we just did, the female's genotype is big A, big A. The male genotype in that case would be little a, little a. So again, it's just calculating the different probabilities. Now, we're going to look a little bit more at laws of inheritance. Gregor Mendel was one of the forefathers of genetics. He came up with something called the law of segregation. This means that the gametes are going to contain one set of the genes. So if you're giving away a sperm, it's only going to contain one set of the genetic information. Additionally, there's a law of independent assortment, and that says that the genes assort independently during gamete formation, or there's no real rhyme or reason as to whether or not a particular gene gets into a gamete. It is completely independent of other genes. Now, with gene expression, I mentioned a little bit earlier that there was a dominant and a recessive type of gene. The dominant gene we're always going to write with a capital letter. This is the phenotype that is expressed. This is what you're going to see. So if you're carrying a dominant gene for being tall, you're going to appear to be tall. On the flip side, the recessive gene is going to be a masked gene, or in other words, you don't see the recessive gene in the person. This expresses only in the homozygous condition. So in the example that we just looked at, a homozygous recessive gene would be little a, little a. The recessive gene is in fact written in lowercase, and again, we can have a dominant versus a recessive, say in a straight hairline versus a widowed peak, or in other genetic situations like cystic fibrosis. Now, when we look at the way that genes combine themselves, we can have something called incomplete dominance. That's when our phenotype is intermediate. Recall that the phenotype is what we're going to see. So the example is a palomino horse or pink flowers. So if we take a white horse and a brown horse, we get a palomino horse. If you're not familiar with horses, something that's easier might be a red flower crossed with a white flower will give us a pink flower. Now in codominance, that means that both of the alleles are expressed equally. An example of codominance would be blood type. The blood type AB is expressing both type A and B alleles equally. And another codominance example is in sickle cell anemia. This picture here shows you the situation with the palomino horse. So we've got the white horse with the brown horse or the chestnut horse, and we end up with an intermediate color or the palomino. And this picture is of the red blood cells showing some sickle cell anemia. All right, we can also have something called polygenic inheritance. Polygenic inheritance is the inheritance of phenotypic traits dependent on many genes. So not as easy as saying it's either a dominant or a recessive. There could be multiple alleles in place here. Eye color, for example, has 3+ genes that are involved in giving us the eye color. That should make sense, however, because the variation in eye color is significant. There's very many shades of brown, many shades of blue, for example. Other traits that would be polygenic are height, body size, and body shape for that matter. Taking a look here, you can see some of the variation in the eye color just between greens and browns and blues. Now, we also have a situation called linked genes. This is when genes for different traits are located on the same chromosome. They create what we call a linkage group. That linkage group then gets inherited together. Sometimes this doesn't work perfectly well because of what we call crossing over, or the swapping of genetic material between different limbs of a chromosome that happens when these cells reproduce. And then there's also some other sources of genetic variability. Independent assortment of genes on different chromosomes gives us some genetic variability. Crossing over, as we had mentioned, swapping some genetic information between different legs of a chromosome. And then random fertilization of an egg by a sperm. These are all reasons that you're not going to look exactly like your sister or exactly like your brother.